Hi, welcome to today's Journal Club. The title of the paper that I'll read today is Chemically Induced Reprogramming to Reverse Cellular Aging. And this research was done by David Sinclair Lab in Harvard with Dr. Jihin Yang. And this is a key slide that you have to know. All you have to know is within this slide, and here is the key schematics. On the left side is the young cell, and on the right side is the old cell. And as the young cell undergoes aging, it shows this uneven or unclear nuclear compartmentalization, which is NC, as the nuclear elements goes outside into the cytoplasm, and this is a well-known hallmark of aging. So what the authors actually just did was to treat these cells, old cells, with just OSK alone, or the chemicals which actually mimic that to show under the image of the cells have this now well clear boundaries for their nuclear or NC. And when they actually looked at the functions, when the cells underwent aging, it has decreased inflammatory signatures and increased cellular respiration, or I think this is related to the cellular metabolism. And you can see, so to summarize, the VC6TF or C6YSNYSA, this is typo, are the chemicals which can reverse aging by mimicking OSK effects. And the reverse fibroblasts actually showed restored NC as well as younger transcriptomic age, which is done by machine learning models. You can see the younger cells exhibited increased cellular respiratory and decreased inflammatory signatures. They've actually tested this with six cocktails, and it's really just essentially just two of them with plus the boosters such as sodium butyrate, which can increase the pluripotency. And when they actually check the transcriptomic age by machine learning, they only show that this compound, the VC60F, was most effective with 5 years age younger. And the small molecules they've chosen were not actually found by themselves, but they actually brought it from the papers like this, which actually showed before that you can actually program the cells chemically. And the further research that we have to actually do is that all the papers that went about pluripotency and making the stem cells was focused on finding the function of OSK itself and as well as the chemicals which can replicate the functions of OSK. So it's more kind of upstream of this regulatory hierarchy. But in order to actually well control this age reversal, controlling the age itself, we have to actually know this hidden layer, which is actually regulated by OSKs. So OSK are the transcriptional factors. So what we think is that there's about to be a some kind of network system which is more precisely targetable than just targeting OSK, which is really upstream, so it could cause, for example, cancer or well known as teratoma. And that was it. Thank you.